Today, understanding complex living systems is hard, but does it have to be? I'm Markus, I'm a biologist.
I'm Phil, I'm a statistician. Together, we discuss the best experiments to cut through biological complexity. Join us to explore the shape of the next experiment.
Hey Phil, we're back again.
Hello.
For this episode of the next experiment, I'm really excited. We've got Rob Howes on, so the most senior person we've had on so far. We've had a lot of early career researchers so far, and Rob is head of biology and DMPK within discovery at Charles River's laboratories. And I think what was particularly cool about this discussion is that he's obviously got quite a long experience of both using multidimensional methods himself, but then also promoting them and rolling them out within various different organizations.
Yeah, it's interesting to hear that you've been using JMP for longer than I have, so that was something. I also enjoyed hearing about, we made this connection between assay development and production processes, right? So we're trying to do the same thing. We're just trying to learn about the process as fast as possible to make sure that the process is robust. That applies whether it's making shampoo or whether it's making a robust assay that you're going to use for drug discovery. So that was great. And I think a lot of people will be very, very reassured to hear that they're not going to be replaced by AI robots.
Yeah, Rob was pretty emphatic about that, wasn't he? We covered all sorts of things from, getting people to use DOE more, the kinds of things that you need to overcome to make that happen, the kinds of insights that DOE can give, all of the different potential benefits from using it that he's seen as he's gone through his career in different organizations using it. And then, ended on AI and the fact that actually it just means we get to do more cool biology.
Yeah, great episode.
All right, so welcome, Rob. So Rob is the head of biology and DMPK at Discovery at Charles River. We've been working now with Rob for many years, very fantastic advocate for multidimensional experimentation. So we're really excited about having a chat today.
Nice to be here. Nice to meet you both again.
Yeah, well, it's nice to meet you for the first time, I think. I don't think we've had a chance to chat before, so I'm really looking forward to this and finding out more about your experiences, your stories about using multidimensional experiments. And to get us kicked off, when did you first hear about DOE, Rob?
So in the dim and distant past, when I was actually in the lab, I came across it kind of early 2000s. So I had an academic career, did a couple of postdocs, and then I joined a small structure-based drug design company just outside Cambridge, and I was in their oncology group, then moved into their screening groups. And it's whilst I was doing that I came across DOE as a methodology. I'd been traditionally trained, like most people, to do one factor at a time. It was okay, but it was slow and tedious. And because a lot of my work then in that team was around generating new assays, things that just didn't exist before, I was getting frustrated at the kind of inability to not just develop those assays quickly, but also if things started to go wrong, how do you troubleshoot it? And, just going through endless cycles of changing different reagents just became massively frustrating. So all of a sudden, I came across DOE and thought, hey, this is great. This is this seems like a really good answer to the ways of doing it. So JMP is the first package that I was introduced to, and it was great. It's really good about stepping you through the process and about getting that ability to start looking at the various factors in a single experiment. So I used it. When did I use it? It was probably like 2005 was the first time I used it. It was
wow, it's before me. I wasn't using JMP then.
Welcome. Do I get a medal or something? So we had a project. We were trying to develop inhibitors to HSP90. That was kind of the project, and we also had a kind of spin-off project about inhibitors for HSP70. And that HSP70, it was a fluorescence polarization assay that I was developing that I used design of experiment on first. And it was, it was
Right.
great. I'd already set up the HSP90 assay, and I knew how much work that took to get set up. And then all of a sudden, this one was set up, but also the real benefit for me was I understood the parameters then that were, sensitive or insensitive within the assay, so that I was able to change conditions, not have, minimize certain reagents, make sure that we had the most optimal robust point within that assay.
And do you remember, given that you'd essentially kind of run the control initially, so it sounds like you did your HSP90 or OFAT and then HSP70 with DOE. Do you remember the differences in the timelines and the progress and that kind of thing, both in terms of, the amount of time, but also how it felt?
So the HSP90 one, it was kind of co-developed between me and someone else, and that took us a decent amount of time, to develop that. I think it was several weeks. The HSP90 one, essentially the hardest part for me was doing the pipetting. It took me a week to do that whole, to basically get the concept of DOE, understand the package, design the experiment, run it, analyze the data. That was probably a week, 10 days as a new user.
Wow.
Yeah. Fortunately, there were some other people in the organization that had used JMP previously. So I was able to lean on them quite heavily, because otherwise it could have been a bit intimidating. But, that was a pretty fast timeline to be able to do that first experiment.
Yeah, definitely. Can you remember, why did you end up using DOE on that project? Did somebody come to you and say, look, the way you're doing this, Rob, is just madness. You really need to, there's a better way of doing this.
Yeah, you're an idiot, Rob.
Maybe just block that bit out. I don't remember the nice things they said to me about it. I think it was just that, here's a different way of doing things. I always like to look at things that are slightly different, novel ways of doing something. And it always struck me as slightly bonkers when we're doing assay development that we're just tweaking a single factor, and it means that your cycle times can take forever. So when you come across the DOE methodology and just think, well, hang about, so I can do multiple factors within the same experiment and get to that point really quickly. Why wouldn't I try it? So it was more a case of suck it and see. So one of the nice things about that company, so the company was Vanalis, who are still based over at Grant Park. And the great thing about that company is they kind of gave you that freedom. Just go off and try it and see if it works. And, I look back on the team that we had around us, and we've all gone on to do very interesting things. And I think that was a really nice culture that they embraced over there, about just try things to see if it improves what we're doing currently.
Yeah, I think that could be really important. And did you have any hang-ups? Like when you first, someone first started explaining this to you, and, there's all sorts of things which, are quite different from the way that we'd usually do stuff as biologists. Are there any of them which you're like, oh, hang on a second, that seems a bit weird?
I think it's that bit. The hardest bit for me was trusting the sort of interpreted bits of data, so that, I'm sampling these different points, and then it's making an inference of the data in between those. And, you're trained to almost do every data point. And so that was probably the hardest thing for me, was just to trust that you're sampling the space effectively.
Right.
And therefore you can draw conclusions. And of course, the first time I did it, I didn't really get it in my head that, of course you can do multiple iterations of this as well. So you can do that kind of wide experiment to begin with, but then you might then narrow down into a different area and maybe do a few more closer data points just to make sure that everything is behaving as you expect it to be. So that was really the main thing.
That's kind of funny, actually, because that's the reason that I didn't even try DOE for 7 years after I first heard about it, because I kind of just dismissed it, because it was talking about 2 levels, and I'm just like, well, what can you do with 2 levels, right? I mean, biology is way more complex than that. It clearly doesn't apply to my system.
Yes, we all like to think that our systems are very complicated and special,
and they are, but just not that different,
It gets you to see how. I mean, it's always one of the things I've always liked about doing DOE is that when you can sample that space, you can start to see, are there hotspots? are there areas where you've actually got some weirdness going on? Because, we typically try and drive for biggest signal to noise, assay window, highest Z prime, and that can lead you to weird points in that topography. And actually, we've had a few examples where actually that's not the right place to be. You maybe want to be, if you want something that is more robust, you can still sacrifice your assay window because you might still get the same Z prime value because your data is more robust, it's more reproducible. You're taking some of that noise out of the system.
Yeah, I think that's really interesting. I think I'm kind of guilty of talking a lot about designed experiments with one outcome. We always talk about yield as being the big thing that, it's kind of the obvious thing to talk about. We can improve your yield, but in reality, most of the time we've got multiple things that we need to understand, right? Multiple outcomes that we've got targets for. So in assay development, that's, Z prime is a really good example of a more complex thing that you're aiming for. Maybe you could explain that a little bit more, because I'm not sure if everybody listening would know what Z prime is. I think I've begun to understand what it means, but I wouldn't mind a reminder.
No, it is a concept that we tend to use in the sort of biology assay space, where it's an indication of the quality of your assay. So what it really means, it says Z prime is a value between 0 and 1. And the way you determine that value is you take essentially your 100% signal, so your fully activated signal, and then either you should, in theory, do your 100% repressed signal, so your 0, or actually just your 0 value. Now, those two should be the same, but there can be differences why. So you have basically your positive and negative controls. And what you do is you run a lot of data points around those to understand what's the distribution of those controls, so that you then get a standard deviation measure of those. And your Z prime is essentially the difference between 3 standard deviations of your positive control from 3 standard deviations of your negative control. It's that gap in the middle. So imagine that you've got, a 0 value and 100% value. You've got a bit of wobble around each of those, and it's that gap in the middle is your Z prime, and that's a value between 0 and 1. 1 is perfect. That never happens. But we always look for something in the range of 0.7 upwards, ideally. And, for some of our assays that we run, they're typically well above 0.9. And what that tells you is that your positive and negative controls are consistently at the values that you would expect them to be. Now, if you're going into something like and that's really, typically you see that in a very simplified system like a biochemical assay, where maybe you've got a single protein with a substrate that gets converted to a product, and you're looking for inhibition of that reaction. That can be really a really high value. If you're looking for a more complicated system like a cell-based system, there's a bit more noise in the system, and so your positive value might have a bit more variation around that 100 signal, and therefore your Z value will be a bit smaller. So it's a really good measure of how good your assay system is. So we talk about it all the time. So if somebody comes along to me and says, I've just developed an assay, it's got a Z prime of 0.4, I'd usually go, please go back in the lab and try and optimize that again, because that tells me that that's quite a noisy assay. If you come back and say 0.9, like, that would be great. But it also comes back to understanding some of those other things. What I really want to see is, have you then tested multiple components to get to that value, so that you then understand the variation within that system as well, not just the min-max signal and variation of those.
Yeah.
Right.
And I mean, and from my kind of, in this respect, I'm more like a layperson than not, because I'm not a discovery scientist originally. And my understanding of Z prime is essentially you need a very rigorous signal to noise measure for these assays, because, in a high throughput screen, you're going to be running them north of 1 million times. So your chances of getting false positives, false negatives are very, very high unless you have an exceptionally clean assay.
Exactly.
Right.
So that's a really key point, is we're often looking for things that inhibit that signal, and you need to know, is it really inhibiting, or is it just the noise around that you would get around that particular positive?
Yeah, so I mean, this is really important because ultimately, this, if you don't have good assays, you can't do good drug discovery. You can't find the treatments for these, these diseases that we don't currently have treatments for.
And I think that's that's an emerging feature across the whole industry, is the quality of the data. There's a lot of data out there that's being produced, but actually what we really want to do is to focus on generating high quality data, because then you're using that as a basis for things like machine learning, AI-based models, so that they are operating off the most robust data that you can produce to hopefully produce then the best predictive models. The idea being that then over time, you might be able to replace a lot of that wet work data with actually predictive data. So then you're only having to do a subset of samples within the actual wet lab, gives you more capacity. So, certainly from our perspective as a CRO, that's a really nice place to be in, because say we have 100 slots to be able to do something, well, that might be only one project in the old days, but if we have a really good predictive model, we might only be able to need to run 10 for that particular project. It means that we could potentially then run another 9 projects of a similar size.
Wow. Yeah, that's huge. So I guess we can get onto where this is all going in the future later maybe, and that sounds like a very exciting future. But I mean, going back to the sort of specific challenges of applying design of experiments, high dimensional experiments in biology, what else? We've already talked a little bit about some of the complexity of assay development. What else makes it, biology uniquely challenging?
I think there's inherently, I think we tend not to standardize particularly well in biology.
Right.
A lot of stuff is bespoke, and therefore it's quite hard to get people into a mindset of, actually, if you do this the same way, you'll get much more reproducible data. And I think the fact that it isn't generates there's a lot of that noise and variability. And I think that's a real challenge for people to then move into the sort of HDE space. But actually that's almost the reason to do it, is because it allows you to understand that space and understand what are the factors that are causing that noise within your particular, whether it's a process or an assay. For me, that's probably one of the main things about biology itself, is probably more the way that we use it. I mean, they are complicated systems. Often, we're often using relatively stripped back versions of those systems. So I think that simplicity actually really applies itself to HDE as well.
Right. So biology is complicated, and the way that a lot of people experiment within biology, they're just making it more complicated. And so DOE is a part of the route towards simplifying things and standardizing and getting results that you trust and better quality data.
And I think because of the way that we would tend to do a single factor change previously, you're not exploring much biological space, so that you're maybe not finding the optimal conditions for your particular system. And that can be a real challenge. Whereas one of the benefits of obviously HDE is you can sample a much bigger space and really understand, am I meant to be driving for that higher signal, or is actually that getting me into a place where, yes, it might be a very high signal, but it's quite fragile. And what I'll end up with is a lot of failures within my system. And actually, I'd rather have a system that is more robust, one that I can then, productionize at a much larger level. As you were saying, Markus, if you're doing a high throughput screen, you could be screening anything north of 2 or 3 million data points, and you've got to have a really complex and a really robust system at that point.
Yeah, it's funny when you talk about discovery in that way, and you just use the term productionize, and I think that's a really revealing term, because we often think of these kind of methods, multidimensional methods, they're often used in optimization for manufacturing. They're often used for, like I first came across them in process optimization, so bioprocess optimization, producing more protein from a system at a higher quality. But really, this is you're doing the same kind of thing. It's just for a different system. You need to make a process that's just as robust as any manufacturing system. An assay that might have to run north of 1 million times in that high throughput screen, but then might be run for a decade hence as that medicinal chemistry is done, refining those leads that are found in that high throughput screen as you go. So I think it was quite interesting coming into discovery and seeing that actually a lot of it in a lot of places is still run in a relatively kind of low, in an OFAT and kind of iterative manner, right? I almost said in a more academic manner, but that isn't quite exactly what I'm looking for.
No, I know what you mean. No, I think it's a challenge depending on the type of organisation you are as well. We're quite lucky in that we're a large CRO. We typically would do 100, 150 plus assay developments or transfers every year in the groups. Now,
Wow.
There's a real demand there to have a really robust system for making that a slick process. If you're in a kind of startup biotech, you might only be running 2 or 3 assays, and therefore there may not be the demand there. But of course, if you don't embed this in the culture at the beginning of an organization, it's really hard then to bring it back in later. So personally, we'll maybe come back onto this, but that's been one of the interesting observations I've seen within Charles River has been around the uptake of this technology within certain parts of the organization who take it up better than others.
Yeah, I mean, like we're starting to kind of move into change management here, and I think that is definitely I think if there's ever anything that's holding back the way we're doing experiments in biology, we know the methods, we know they work exceptionally well in a lot of instances, especially in this area of discovery where we can do a lot of individual runs, we can do a lot of wells simultaneously, lots of different conditions. So we know it works exceptionally well. And yet, as we know, it still takes a fair bit to actually instill this as a way of working and for that to spread and to hold within organizations. And, you've had the experience of working in multiple different organizations now and seeing this technology or this way of working in lots of different contexts. Have you been able to glean some key things that you think about, what we can do to get people using more of these kind of multidimensional methods and keep using them?
There's a few things that I've kind of noticed over time. So going back to my own personal experience, it was always a struggle to do the wet work. So to actually use the software and to do the design and then the interpretation of the data, great, that was there. One of the challenges was always about just that really complicated experimental setup you'd have to do when you're trying to do the theory experiment. So obviously that's now not a problem with Synbase, and having integration of that into the dispenser. So that's great. That takes away that barrier. I think a lot of it does come down to individuals. So within large organizations, I've seen this both at AstraZeneca, where I worked and helped to introduce design of experiment in there, but also over at Charles River has been around. You obviously have pioneers, people who they get it, they're really passionate about it. They can be the people that really understand the technology and can help train others. I think then you get into a really interesting place around what groups then within your organization are really good at taking this up. We found some of the best groups have been the young scientists, so those early career professionals. We tend to work with our fresh graduates and apprentices and just almost from day one, just say, this is how you develop an assay, so that they're not thinking about, they're not having to unlearn another way of doing it. They're just like, well, of course, this is the way I would do it. This makes perfect sense. Why would I do it any other way? More of the challenges we have are maybe some of those more people that have been doing this kind of OFAT for quite a long time. But that's kind of a slightly unfair comment, because that's only because of the challenges of the types of assays we're trying to build. So you kind of referenced this, Markus. We have a range of assays that we might build. So a lot of them might just be a straightforward this is for a short-term project. We're not going to use it ever again. Okay, fine. Maybe that's where their more traditional approach might be acceptable because you might be inheriting a relatively robust assay. But actually, most of the time, our assays, we certainly use them for 6, 12, 24 months. And often it is something that is done at a large scale first. So 500,000 plus data points as that large screen. And then it moves into a more of a profiling setup where it might then stay around in the project for 24 months. And actually, one of the things that we'll need to do as well is when you're going through the optimization phase of a project, you often hit the bottom limit of an assay. So the limit of sensitivity. So we've also used it to then re-optimize those assays to make them even more sensitive. So that was one of the things that I did when I was at the bench with our HSP90 assay. That assay had kind of lower limits, and as the compounds that we were developing were getting more and more potent, we had to then keep re-optimizing that assay. And so DOE was a great way to do that. Just say, well, I kind of know the conditions, but let me just explore a few other things at the same time, and that allows you to get to that. Even the same assay gets multiple variations, get to that variation a lot more quickly.
That's really cool. So you're essentially zooming in. Your compounds are getting so good that you can't differentiate between them anymore.
Exactly. I think our first assay, I think it had a bottom limit of 50 nanomolar. Compounds soon hit below that. Then we had to go down to 10. Then we ended up with our final assay was a kind of sub-nanomolar sensitivity, which was great because the compounds we had were low nanomolar.
Huh.
Yeah.
That's really cool. So
yeah.
So when we're thinking back, I mean, you've obviously had a fair bit of experience now, in terms of seeing who adopts things best and driving things forward, changing cultures of how people are doing experiments within organizations. Along that way, you must have seen a lot of different multidimensional experiments run. Is there any amongst those, be it a success or a failure, that you really felt that you've learned a lot from, that sort of stands out in your memory?
I think the one that comes to mind is one that comes from Charles River, and I will give all the credit to Gareth Langley at Charles River, who is the real pioneer within that organization. And that was one where we were looking at a particular assay of, they were looking at a cofactor and the concentration of membrane. And what we'd done is, this was a really good example of, they'd driven the assay to a really high signal to noise, but it was a bit flaky as an assay. And so, we were getting a fair number of assay failures. And so that's not great, because especially when you're in a CRO, trying to do things right first time is a really important metric. So what they did is they used HDE to actually look at different factors and found that within the sort of topography of that assay window, there was actually another part where it was instead of it being a peak of response, there was a much larger surface of response. That wasn't quite the same. It wasn't quite the same signal to noise, but actually it was a lot more robust, and that was a lot better for us in terms of being able to then move that assay into that more robust space. And it was just a really good example of something that you hypothetically, don't often see it in reality. And it was nice to actually prove, though, that yes, the reasons that we were getting these assay failures was just we'd originally, using OFAT, driven this to a signal peak, but that signal peak was quite fragile.
Right. So in the original experiments, you were seeing really good Z primes, but then just some kind of noise factors that you weren't accounting for when you put it into production, as we were saying, meant that it would fall over too often.
Exactly. So you can still have a good Z prime in terms of your assay when you run it, but if 3 out of 10 plates fail because there's just no signal, then that's really poor. So, and you can imagine, take that to the example that we talked about of a high throughput screen. if suddenly 30% of your plates fail, then the cost and the just the effort required to rescreen those is huge. So what we really want, ideally, is something where we have a really robust assay that we understand all of the factors that go into making it robust, but also something that we can do right first time, and so we can run it at that scale. And then it makes it a lot easier then to do, really push the optimization of that particular assay. So in the assay world, most people, I think, tend to work in 96 well plates traditionally.
Mm-hmm.
As a minimum, we tend to work in a 384 well plate. So already you're driving miniaturization of your assay. When you're looking to do something like a high throughput screen, you're really talking about a 1536 plate. So you're really miniaturizing your assays, and you're really trying to have to optimize that with a much smaller volume, very different, can be very different conditions within that 1536 versus a 96 well plate.
Right. And the liquid dispensers are getting really good, right? I've seen some of these amazing technologies in Markus's lab before, but still, I mean, if you're scaling it down and down and down and down, the relative variation of concentrations that's just uncontrolled, that's getting more and more challenging, right? You're just going to have more and more noise in the amount of the different reagents and ingredients that you're putting in there.
And when you're when you're talking about things like high throughput screens, you're then starting to look at not just your kind of Z prime per plate, but how do your controls work across multiple plates? Do you actually have variation within a plate? So one of the classic things you get is edge effects, especially with things like a cell-based assay, where maybe the temperature and the gas concentrations are slightly different to the center of the plate, therefore your cells might respond differently. All of these factors, you need to try and minimize any variation. And, trying to do that one factor at a time can just take too long, and potentially drive it crazy as well.
Yeah.
Yeah.
So there's just more and more things that are sort of, this increasing miniaturization is driving like more and more challenges. Like there's an even greater need to be precise and accurate in your experimentation.
Yeah, and I think what's nice, it kind of forces you into it as well, because once you get into 96 well plates, you can do by hand, and so you're still quite connected to it. Once you get to 384 upwards, you can't do it by hand, right? So you've got to then use automation, use dispensers, and that almost gets your mindset into, okay, well, if they can do that, what other things can they do, and how can they help me really optimize my assay?
That's really interesting, actually. I didn't think about that kind of driving force in quite the same way of miniaturization, because when I came into the space, the thing that really excited me about the miniaturization is that, I come from a place of bioprocess development where maybe you can run a dozen different conditions, right? And so then you come into discovery, and here you want your final process to be in a 1536 well plate. So that means you can do 1,500 different conditions. And, the total volume is 10 mils or something? I mean, it's just completely
unimaginable.
It's completely off the chart. So I've always seen it as a massive opportunity to be running, multi-dimensional experiments in a way that I had never imagined possible. And frankly, I don't think has been applied in any other field. It feels like a completely new area. We've been using the terms, DOE, HDE relatively interchangeably. But I think that high dimensional experimentation is what, I'm seeing as a kind of evolving term, which is like a subset of like these multi-dimensional methods, but is one where it's taking advantage of this miniaturization and automation to really kind of do these exceptionally expansive experiments. Which I think is just a fascinating area to explore. And, you guys are so used to this, you're spoiled, right? You're used to being able to run, huge numbers of runs, but, coming in from where you can do amazing things with like 32 runs, right? That was my first year. We was 32 runs. And so, the idea of being able to run, pretty much 2 orders of magnitude more than that is just insane, right?
Yeah, maybe we are spoiled, but that's, we're a data heavy part of, I guess part of science that we expect to be able to generate that much data, but it does allow you to then really, push your optimization. And one of the things that I really like about it is you can optimize in many different ways. So, we've talked about signal to noise and robustness. And, those are kind of fairly typical things that you might want to optimize against. But actually, things like sustainability come into it. There's a really interesting angle there about, well, can you use this to minimize the effect of, or even remove certain toxic reagents in there? Or just generally by miniaturizing, you're then reducing the amount of reagents that you're using. So one of the things that we talk about a lot internally is, when we are looking, we need to be clear when we're going into an experiment what we're trying to optimize for. And it's very easy for people to then come up with 20 different things to optimize for. I'm like, no, you need to, what is the key thing that you need to optimize for? But it can be really different, it's one thing I find fascinating about the methodology is it's not just it's this one thing that, as you described right at the beginning, it's not just about yield, right? It can be many different things. Because again, one of the issues that we have in the discovery space is we use a lot of plastics. So one of the things we're looking at is, can we try to use this to reduce the amount of plastic that we're using? So there's a really strong and increasing sustainability angle that's starting to permeate because of this methodology, which I don't think I would ever have predicted, 10, 15 years ago.
No, it's definitely, I mean, in the last 5 years, across all the industries that I've worked with, that's increasingly, it's how does this, our company goals and now, one of the top 3 is about sustainability. How does this play in sustainability? And obviously, I think there's the obvious, you can do more efficient experiments, and experiments have a cost to them. So if you can do them more efficiently, that's more sustainable, right? But I think the bigger impact probably is in helping you to develop new processes that are inherently more sustainable, as you say, like learning faster about what it takes to replace some of these toxic reagents or some of these undesirable plastics that you're using.
Yeah, and that's one of the, it's when I was a postdoc in the States, I worked with a guy who was just like, look, I try and replace everything with water. It's a really nice way of thinking about optimization. Why do I need this thing in my particular system? And the great thing about this is you can then just go, okay, if I leave it out, what effect does that have? All the way through to, multiple times the kind of expected concentration. Well, if you did that one factor at a time, you probably would never do that experiment. Whereas obviously DOE allows you to explore that space. And, where you really want to get to is, yes, I've got the minimal number of components in my system because it still makes it robust. It's still the right biology, but I'm not then having any excess stuff in there.
Well, not often it can be more robust, right? Because like every addition, every extra bit of complexity adds to that lack of robustness. So we've seen this particularly in the cell culture space, where people are using these more sort of sophisticated cell-based systems. Then often the methods will be pretty arcane, this really kind of rich and complex cocktail of different growth factors or whatever, which have been established over the years that you need all of these things, but only if it's a gibbous moon and that kind of thing, right? And then we've seen multiple times that actually you run a more multi-dimensional experiment on it, and you can often remove swathes of these additions. So you end up with something is, dramatically simpler, dramatically more robust. And I think from a sustainability perspective, like actually, you spoke about it earlier, Rob, having a system that doesn't fall over, right? Because that is literally a waste, right? You couldn't get any more wasteful than that. And so having a system that's less likely to fall over and quicker to stand back up again when it does, right, is something that I think, we often have this problem of properly articulating the value of this. You've almost got too many things you can point at. We're getting better quality data, which has to be a key thing. You referred to it earlier, Rob. AI ready data. It's incredibly important, and it's amazing that it's taken AI to come along for people to focus on the quality of data. But that's a side point.
I think they have.
I know. I'm being facetious.
Yeah, I know you're being facetious.
and then obviously there's time savings, which is not just actually that you're moving faster in that project, but all of those resources that would otherwise have been used, for that lab space and that time, the full economic costs, and then cost savings of reagents as you strip them down, savings on robustness, so you don't have to stand things back up again. The sustainability angle, it's almost too much, right? Like it's and it sounds too good to be true a lot of the time as well. And you have to really walk people through and say, no, we're not snake oil salesmen here. It's just because this is a much better way of investigating these kinds of systems, and you kind of run out of persuasion.
No, I know what you mean, and I think there's other ones that I'd put in there. So for some of the things that you do in the discovery area, you've almost got an ethical responsibility to get it right first time.
Right.
If you're doing an experiment that is using the product of some in vivo system, right? It's incumbent on you to make sure you've got a robust system so that it's not falling over, and therefore using design of experiment to really build in that robustness. It almost feels like an ethical obligation on you as the scientist in the lab, because then you can minimize the number of in vivo animals that are used, whilst we're then going through the process of developing obviously alternative systems that can replace those. And I think the other one that is an area, you were talking about cell culture. I think one area that I can see a really big benefit for this, which we're sort of dipping our toes into, are really complex cell type generation. So there's obviously a lot of work. We do quite a lot of work in that generation of iPSCs that differentiate into different tissue types. So these are really...
Sorry, what's IP?
Sorry, I now realise I've gone back into lingo. So these are induced pluripotent stem cells. So...
Oh, yes, we've talked about these before, yeah.
So,
getting it.
Cells which have been kind of de-differentiated back into their kind of stem cell phenotype. And the aim is that you can keep those growing, and when you then give them certain factors, you can differentiate them into different types of cells. So skin, liver, nerve cells, just depending on those kind of reprogramming agents that you give them. Now, that's a really complicated, long process to do that. So it might take you 5, 6, 7 weeks to actually differentiate from an iPSC into a particular cell type. And even when you get to the end, it may not be 100% of that particular cell type as well. And I just look at that and just go, that's great that we can do that, but boy, is that ripe for DOE to really optimize it, because then you can understand which factors when will actually give you, either more of a particular cell type or a more robust cell type, something which is more biologically relevant because it's a bit more mature. There's lots of factors that you could look to optimize it against, but it just that really feels like a process which is ripe for addition of this.
Yeah, I've actually seen some evidence of that actually, and hopefully that we can have another guest on later on. But there's Tim Ashlin over at GSK who's been doing some cell differentiation, and these results were presented at a conference last year. And what was fascinating there is that, the vast majority of the literature all said that, to differentiate this particular type of cell, you have to use these particular growth factors. And then there was one paper, sort of one lone paper out there, if you go digging, that suggested, no, it's not those ones. It's potentially this factor. And in a single experiment, he disproved a vast swathe of the literature, and showed that, it was this lone voice out in the wild that was correct. I think I'm hugely excited about the possibilities of exploring these complex cell-based systems. And ideally, because, the end goal there is to be using the right kinds of cells for getting, as relevant data as we possibly can, ultimately to the clinic, right?
And also then starts to lead into things like cell therapies where, you are then looking at both quality and yield, because you're trying to, you're running these things at huge scales. So, we tend to, in the discovery space, tend to work at relatively modest scales. But obviously, as you go further into preclinical and manufacturing, if you're developing a cell therapy, that ability to then get those cells of the right type at the right scale is absolutely critical.
Yeah. Right. Yeah, because these things are massively costly currently, like, millions or at least hundreds of thousands of dollars per treatment for a patient, for a single patient, right? So it's a huge imperative.
And in the when you're doing a manufacturing run, there's a huge investment there. It's probably in the millions of dollars per run, and you want to make sure then that you're getting not just a high yield, but because it's not just a small molecule or a protein, it's a cell, it's a lot more complicated. So you need to make sure that you've got the quality as well as the yield.
Yeah, of course, yeah. So, I mean, I guess hopefully with the ever increasing complexity of these challenges, then people are only going to be successful if they're doing good experiments, right? So this is going to be a great advert for the power of high dimensional experiments, DOE, whatever we want to call it.
I'd certainly hope so. I mean, Rob, I'd be really interested in, because obviously there's a huge amount of talk at the moment in our space around AI, and there's kind of lab in the loop type talks and all these kind of things. How do you see all of these different, because I am exceptionally excited about the future of machine learning, AI, and that being applied to our field. But I think that one thing that's not clear in a lot of people's minds is exactly how these different approaches and technologies might dovetail together in future. What's your kind of emerging view on that?
Certainly in terms of design of experiment and AI, I think it's a fundamental that you have really high quality data. We've had a lot of discussions internally that do we actually go with data that we've had previously, or do we just generate something which is a really high quality data set from something that's been something that's been optimized, and use those as the training sets for your AI models? And I think that's where focusing on the data quality is absolutely critical. And that comes back to how do you get there? Well, I think the best solution that we have to date is using design of experiment to really understand what's the most robust data set that I can generate in this particular assay. And I think for me, that's where having this as an underpinning, I would expect all of our, where I want to get to within our division is every assay that we do has that almost design of experiment quality stamp on it to say, look, this has been optimized, therefore you can trust the data that's coming out is high quality, and focus on that. Because in the one thing I always talk to my teams about is you're always trying to balance quality, cost, and speed. But actually, especially when it comes for AI, you really need that quality to be there at the beginning, because if you don't have the quality, it's going to be very hard for it to generate a predictive model that's any good. It might only be a predictive model which is very narrow in nature. I think if you want something that is going to be a broadly applicable predictive model, you really need that high quality data. And I think that gets really important when you're looking at areas such as the sort of DMPK space, so drug metabolism and pharmacokinetics, where you're trying to understand, you're understanding how is a molecule processed in the body, and that works across every drug program. So getting a really high quality data set there might give you the ideal situation where you have a single predictive model. So no matter what the molecule is that comes in, you can predict how that's going to behave in a human microsome type format without actually having to do the wet work. I think where we are currently, we're doing more local models that might be more to do with particular compound series. But I'd love to get to the point where all comers have such high quality data sets that we've generated to make the model that you could actually almost get away with doing any wet work, or just do a tiny amount of wet work just to keep training, reinforcing the model over time. So I think, summary, right? I just think it's critical as we go into a world where AI is going to play a really important part in supporting what our scientists do, is about getting that really robust, high quality data.
Yeah. But it's not going to replace these scientists, right? I mean, you're kind of talking about replacing a lot of the wet lab work. What does that mean for the biologists that are currently doing this?
So it's augmenting what people do, right? Why would you spend your time in the lab doing a relatively simple assay, right? That, yes, it generates data and it's kind of easy, but actually, why would you run every compound in a program through that? If you only need to run 10% of that, what that means is we can run 5 times as many projects. And it also might mean that we can run the projects faster. Bear in mind, the whole point about what we do is about generating medicines for patients. So, we're limited by the number of people that we have in organizations. You might have automation that we have. So how can we use that even better so that we can generate more therapies? One of the things that we do a lot within like Charles River is there's a lot of work around rare diseases where you might only have a very, very small patient population, and speed is critical at that point, right? So if we can use this to say, well, we can now take on more of those projects because we've got the capacity, because we're not having to do as much wet work per project. It doesn't mean we're replacing the scientists. It means that we're doing more work and we're bringing more medicines to the, to the clinic. And the cost and the time to get each medicine to the clinic will hopefully be reduced as well.
Yeah. My mental model for this is that the scope of what we can do with biology is essentially infinite. Like, I mean, it's not infinite, obviously, because, nothing is. But like, that's my mental model.
It's as good as, correct?
And, yeah, exactly. As good as makes no difference. And so if you take that as your premise, then, we're not going to run out of any biology to do. There's always going to be more, and there's always going to be more good that we can do with it. And it gets me hugely excited to think of, the massive productivity gains that we could be getting through a combination of all of these different methods.
Yeah, and I think it is a really clear vision in my head that you have the ability to do more, not that you're going to replace people. That doesn't make any sense.
Oh, why would you want to replace brilliant scientists? So just, more science with the same number of scientists.
Yeah, which makes it a damn sight more exciting for them too, right? So.
Yeah, well, that's what I think. They'll be doing really interesting work, multiple projects, more things getting through to the clinic. I mean, anyone that works in discovery, the reason that everyone can get out of bed highly motivated every day is every day you're working in the lab, you're pushing a bunch of new medicines closer to the clinic to treat patients. That's a great motivation every single day.
Yeah, that's a fantastic vision for the future. Listen, Rob, thanks so much for coming on. Really enjoyed chatting with you. We'll see all you guys next time on the next experiment.
Thanks.
Lovely to be here. Thank you.